Hello, welcome back to my YouTube channel, and in this video we are going to look at insulin signal transduction. We are going to understand what happens inside the cell when insulin binds to insulin receptors. And this is very important because when we're talking about how insulin regulates glycolysis, glycogenesis, and lipid metabolism, it's very important to understand what I'm going to explain in this video. So, when insulin is released by the beta cells, it goes to its target tissues. And what are these target tissues? So we have got the liver tissue, adipose tissue, and the muscle tissues. These are the main tissues where insulin goes to. So when insulin goes to these tissues, what is going to happen? So in these tissues, let's say the muscle cell. In the muscle cell, we have got insulin receptors on the muscle cell. So what you can say here is an insulin receptor. And the other thing that you need to understand is that the muscle cells and the fat cells, they have got a type of growth transporter that we call growth 4 transporter. And then the liver and the beta cells, they have got what we call growth 2 transporter. Now, other tissues, they've got also their own types of growth transporters. But the growth 4 transporters, they are specifically for the muscle and the fat cells. And what you should understand is that the GLUT4 transporters, they are the only type of growth transporters that are insulin sensitive. The rest are not insulin sensitive. But why is it like that? So it's important that you understand this. So the GLUT4 transporters, they are found in the cytoplasm of the muscle and the fat cells. But the GLUT2 transporters and other growth transporters, they are found in the cell membrane, embedded already in the cell membrane. So the GLUT4 transporters, they require insulin for them to move from the cytoplasm to go and bind to the cell membrane. While the GLUT2 transporters and other transporters, apart from the GLUT4, they don't require insulin because these are already in the cell membrane. So here, let us imagine that this is a muscle cell. What is going to happen is that insulin is going to come and bind on the insulin receptor. Now, the insulin receptor, it has got 4 subunits, as you can see. So this is the insulin receptor with 4 subunits. So we've got 2 alpha subunits exposed to the outside, and we've got 2 beta subunits embedded in the cell membrane. Now what you should know is that the beta subunits, they have got an intrinsic tyrosine kinase enzyme, and they have got a lot of tyrosine amino acids. So in the beta subunits, you should know that there is an intrinsic tyrosine kinase enzyme, and we have got also tyrosine residues, or in other words, tyrosine amino acids. Now, the alpha subunits, they provide the binding site for insulin hormone. So what will happen that when insulin comes, it's going to bind to the alpha subunit, and then it's going to activate the beta subunit. So the beta subunits, when they're activated, it simply means that the enzyme inside the beta subunit is activated. What enzyme is found inside the beta subunit? We said it is tyrosine kinase. So tyrosine kinase is also activated. Now, kinase enzyme, they phosphorylate other molecules or dephosphorylate other molecules. So tyrosine kinase, what does it do? This is an enzyme that phosphorylates tyrosine amino acids, or in other words, tyrosine residues. So tyrosine kinase enzyme, when it is activated, is going to phosphorylate the tyrosine residues that are found within these beta subunits. So when the tyrosine residues are phosphorylated, these 2 beta subunits are going to come together and dimerize. So what do we mean dimerize? They are going to bind to one another and form one structure. That's what we mean. So what you can see here, these are phosphate groups. So this is simply showing that we've added phosphate groups to the tyrosine residues. Or in other words, so when the tyrosine residues are phosphorylated in the beta subunits, it means that this is fully active. And this is what we call autophosphorylation. We have got a protein that we call IRS. So this protein is going to come and bind to the phosphorylated beta subunits. And IRS, it simply stands for insulin receptor substrate. So this is a protein that binds to the insulin receptor. And that is why it is called insulin receptor substrate. So normally, this protein, it has no phosphate groups. But what will happen is that it's going to come and bind the phosphorylated beta subunits, and it's also going to be phosphorylated. So it is going to become phosphorylated because it has also tyrosine residues in it. So IRS protein is going to come and bind to the phosphorylated beta subunits, and it's also going to get phosphorylated. And what you should know is that in the muscle cells, the main type of IRS protein that we have is IRS2, because we've got other types of IRS protein. We've got IRS1, IRS2, IRS4, and others. So the IRS2 is the one that is found in the muscle cells. Now, when the IRS protein is phosphorylated and activated, there's going to come another enzyme. So you can see this enzyme. So this enzyme that we have there, it is called PI3K. Now, what is PI3K? So PI3K, it is simply standing for phosphatidylinositol 3 kinase. And this enzyme, normally it has no phosphate groups. And it has got this binding site. So this binding site of this enzyme, we call it SH2 domain. So this enzyme is going to come and it's going to bind to the IRS protein, which is already active. So when it binds to the IRS protein through its SH2 domain, this enzyme, which is PI3K, phosphatidylinositol 3 kinase, is also going to become phosphorylated and activated. When it becomes phosphorylated and activated, what is it going to do? So in the membrane of cells, and right now we're looking at the muscle cell. So in the membrane of this muscle cell, specifically the inner leaflet of the cell membrane of this muscle cell, we have got a lipid molecule which we call PIP2. So PIP2 simply stands for phosphatidylinositol 4,5 bisphosphate. Phosphatidylinositol 4,5 bisphosphate. This lipid molecule is found within the inner leaflet of the cell membrane of this muscle cell. So now, PI3K enzyme, it is called phosphatidylinositol 3 kinase because this enzyme is going to add a phosphate group at carbon number 3. You should know that kinases, enzymes, they do what? They phosphorylate or dephosphorylate. So this enzyme is going to phosphorylate PIP2 at carbon number 3 so that it becomes PIP3. So phosphatidylinositol 3 kinase is going to add a phosphate group at carbon number 3 so that we form PIP3. So PIP3, it simply stands for phosphatidylinositol 3,4,5 triphosphate. So the conversion of PIP2 to PIP3, it is catalyzed by this enzyme which we call PI3K, phosphatidylinositol 3 kinase. Now when you form PIP3, let us look at PIP3 and see what it's doing. So we have got PIP3 right there. So this is the one that I've drawn right here. So it is having phosphate groups at carbon number 3, 4, and 5. Now, this PIP3 is going to act as a docking site. What do I mean? There's going to come two enzymes, PDK1 and another enzyme that we call AKT. So PDK1, it is simply standing for phosphoinositide kinase 1. Phosphoinositide kinase 1, this enzyme is going to come and dock on PIP3. Another enzyme, which we call AKT, also known as protein kinase B, will also come and dock on the same lipid molecule, which is PIP3. Now, when these two proteins, when they bind to PIP3, what will happen that PDK1 is going to become activated and it has got these legs carrying phosphate groups. So it will start adding the phosphate groups to AKT. So PDK1, after binding to PIP3, is going to become activated and it will start phosphorylating AKT, which is also known as protein kinase B. Now, when AKT is phosphorylated, this enzyme, it is very, very important because this is the enzyme that is going to influence glycolysis, glycogenesis, lipid metabolism. It is this enzyme. So this enzyme is very critical. Now, AKT or protein kinase B, when it is active, what is it going to do? Remember what we said. We said that the GLUT4 transporters, they are found inside the cell, in the cytoplasm. So you can see that here we have got a vesicle, and this vesicle, it is containing the GLUT4 transporters. So these GLUT4 transporters are tricky because they require AKT for them to move and go and bind to the cell membrane. So when you look at this vesicle, you can see some kind of blue dots. So those blue things, those are proteins. So around this vesicle that is containing the GLUT4 transporters, we have got proteins that we call AKT substrate of 160 kDa. So AKT substrate of 160 kDa. So this protein is substrate of AKT enzyme. Now 160 kDa, it is simply telling us the weight of this protein. So the weight of this protein is 160 kDa. So this protein, which is AS160, is the one that binds this vesicle surrounding it. Now, when it binds this vesicle, what does it do? It prevents this vesicle from moving and go and bind to the cell membrane. So it's simply telling us that the tyrosine residues are being phosphorylated. So it prevents the binding of this vesicle to the cell membrane. As a result, it prevents the movement of the GLUT4 transporters from the cytoplasm to the cell membrane. It inhibits the translocation of the vesicle in other ways. So now, this protein, the AS160, normally it is dephosphorylated. It has no phosphate groups. So it's the one that is preventing this vesicle from moving. So what happens is that AKT, or protein kinase B, when it is phosphorylated, it becomes active. It comes here and phosphorylate these proteins that we are calling AKT substrate of 160 kDa. So it's going to phosphorylate these proteins that are binding to the vesicle. Now, when we phosphorylate these proteins, they are going to become what? Inactive. So they are going to be inactivated and they are going to leave the what? The vesicle. So AKT is going to phosphorylate the AS160 and inactivate these proteins so that they leave the vesicle. When they leave the vesicle is going to be able now to move and bind to the cell membrane. So when the vesicle moves and binds to the cell membrane, what is going to happen? What's going to happen is that the GLUT4 transporters are going to bind to the cell membrane, and that will allow the influx of glucose into this cell. So glucose is going to be able to enter into this muscle cell via the GLUT4 transporters. So GLUT4 transporters, they're really special because these guys, they're found in the cytoplasm, unlike the rest of the GLUT transporters which are found in the cell membrane. Now, what is the other effect of AKT? AKT is very important because as you can see, it can come here and phosphorylate this important pump that we have. What do we call this pump? So this pump, it is called sodium potassium ATPase pump. So AKT, it can come and phosphorylate this pump. So when we talk about sodium potassium ATPase pump, we've got two subunits, the beta and the alpha subunit. The beta subunit is the one that regulates the alpha subunit. The alpha subunit is the one that transports sodium to the outside of the cell and potassium into the cell. So the alpha subunit, it's the one that transports these ions. But the beta subunit, it regulates the activity of the alpha subunit. What happens is that AKT, it phosphorylates the alpha subunit. When it phosphorylates the alpha subunit, what's going to happen? This alpha subunit's activity increases. As a result, we'll be transporting more of sodium from inside the cell to the outside the cell and potassium into the cell at a faster rate. So it increases the activity of this alpha subunit. So let me just say that again. AKT phosphorylates the alpha subunit of sodium potassium ATPase pump. And once the alpha subunit is phosphorylated, its activity increases. As a result, we are going to increase in the overall transport of these ions that we're transporting. So the overall transport of these ions is going to increase. And that is very important to understand, because in patients who have type 1 diabetes, when you give them too much of insulin, what happens is that you're going to drive the potassium into the cell. As a result, the potassium in the blood is going to drop, and that is what we call hypokalemia. So, high level of insulin or too much of insulin can lead to hypokalemia, which is very dangerous. So when you're giving a patient insulin, make sure you're giving them a well-calculated dosage. Because when you give them too much, it's going to lead to the driving of potassium into the cell. As a result, the level of potassium in the blood is going to drop. So that is very important to understand. So in the next video, we are going to look at how AKT can activate glycolysis. And we're also going to understand how AKT can activate glycogenesis as well as lipid metabolism. So please make sure you subscribe to my YouTube channel. I'm kindly asking, subscribe, comment, like, and share. I would really appreciate. See you in the next video.